Welcome to Azad Bioacademy. Today's lecture topic is vasodilation. So today we will talk about vasodilation. Usually through our parasympathetic pathway, acetylcholine is one of the master molecule for causing vasodilation. If you think during inflammation, histamine is one of the major molecule for vasodilation. Also, you can think about nitric oxide, which is one of the key molecule for vasodilation. So, in another way, inflammatory reaction and immunological reaction, vasodilatory nitric oxide can produce and it can cause vasodilation. So, in our body, several ways vasodilation can be occurred. Okay, so now we will talk about vasodilation, the basic mechanism of vasodilation, how vasodilation will occur. So earlier I said by acetylcholine, the master molecule of parasympathetic pathway, the nervous system, is one of the pathway for causing vasodilation. So if I consider the pathway as a basic pathway, then another associated influencing ways we will just try to connect it with the acetylcholine pathway. Okay, so firstly, acetylcholine comes from your nerve terminal, that is acetylcholine is a neurotransmitter. Then what will happen? It will work on effector organ. So here effector organ is your blood vessel, because our discussion topic is vasodilation. So this is the effector organ. So in effector organ, the blood in your blood vessel, acetylcholine receptor is there. So acetylcholine is basically show its activity through receptor. So acetylcholine, that is cholinergic receptor. So that can be in two ways. One is muscarinic receptor, another one is nicotinic receptor. And muscarinic receptor has 5 types, M1 to M5, and nicotinic receptors are 2 types, one is NN and NM. So we will not go in that way for today, because our main focusing topic is vasodilation. So come to the point. So if in effector organ, the blood vessel, if I just draw a structure of membrane where a receptor is present, that receptor is, you can draw in that way, that is the receptor. Okay, and this receptor is called, you can see it is the M3 receptor. So already I said acetylcholine receptor is M1 to M5, that is M1, M2, M3, M4 and M5. So in blood vessel, M3 receptor is very rich. Okay, and now you can consider this as a endogenous ligand, that is acetylcholine. Okay, so when acetylcholine bind with the M3 receptor in the blood vessel, then what will happen? This M3 receptor is a G protein coupled receptor. Okay, G protein coupled receptor. So this is the G protein. The G protein is the trimer of 3 subunit. One is alpha, another one is beta, and another one is gamma. So this is the G protein. Why it is called G protein? Because before binding of acetylcholine, this G protein is inactive, because initially GDP is connected with that protein. GDP, that means guanosine diphosphate. From that guanine, the name of the protein is G for guanine. So guanosine diphosphate associated protein is called G protein, and that G protein is coupled with the receptor. So this receptor is called G protein coupled receptor. That means muscarinic receptor 3 is one type of G protein coupled receptor. And the G protein has several types, but among them 3 types are very prominent. One is GI, then GS, and then GQ. Okay, so M3 associated G protein, what type of G protein? That is GQ. Okay, so now come to the point. Acetylcholine bind with the M3 receptor, and M3 receptor will change its structure to capture the acetylcholine. That is, conformational change will occur. And when conformation change will occur, suppose if I try to catch the pain, then I have to just uh, I have to change my finger position, and then your hand position is stretching. Okay, so like that, when the receptor will try to capture the uh, acetylcholine, then conformational change of the receptor will occur, and then associated protein will be activated. How it will be activated? That GDP will be replaced. That is, GDP will just removed from here, and GTP will attached. When GTP will attached, then this GQ protein will be activated. Okay, and you know G protein alpha GQ will be the separated active form, and beta gamma will be another complex form that can be also active, and that those will work on effector protein. Okay, so I will not go to the details like the discussion on G protein. So when G protein will be activated, activated G protein will activate the membrane enzyme that is called phospholipase C beta enzyme. Phospholipase C beta enzyme, which is present in the membrane. Then when phospholipase C beta enzyme will be activated, then here phospholipid, membrane is composed of phospholipid, and also here inositol is there, carbohydrate in nature. So they are present in the membrane. So when phospholipase C will be activated, from there two second messenger will form. One is IP3 and another one is DAG. Okay, IP3 means inositol triphosphate and DAG means diacylglycerol. So here some another step, that means firstly it will break down the phospholipid and it will produce PIP2, that is phosphatidyl inositol 4,5 bisphosphate, and then it will turn into IP3, and phospholipid from phospholipid diacylglycerol will form. Okay, so now IP3, what will happen? This IP3 will go to the endoplasmic reticulum. If we consider this is endoplasmic reticulum, and on the surface of the endoplasmic reticulum, IP3 receptor is there. IP3 receptor, IP3. Okay, so if you think this is the IP3, so IP3 will bind with the IP3 receptor. Then what will happen? IP3, that is endoplasmic reticulum, is basically responsible for post-translational modification, and also responsible for storing intracellular calcium. That means storekeeper of calcium within the cell. Okay, so when IP3 will activate the endoplasmic reticulum, then calcium will come out from the endoplasmic reticulum. Okay, so calcium will increase, intracellular calcium will increase. Then what will happen? This intracellular calcium will just go to the endothelium layer. This is the endothelium layer, and this is the smooth muscle. Okay, blood vessel. This is smooth muscle, and this is endothelium, and this is smooth muscle. Okay, so that calcium will activate one enzyme within the endothelium, that is ENOS. That means endothelial nitric oxide synthase. Okay, that enzyme will activate. That enzyme, what will happen then? When the enzyme will be activated, then it will convert the arginine into nitric oxide. This is the master molecule for causing vasodilation. This is another second messenger. Okay, then what will happen? This is the gaseous state second messenger, and that will be produced within the endothelium. Then what will happen? From the endothelium, it will just come into the smooth muscle region. Okay, so it will come into the smooth muscle of the blood vessel. Okay, then what will happen? Now we can go further step. So smooth muscle is composed of cell. So over there, in the smooth muscle, a membrane, another enzyme is there. That is, if you think this is the enzyme, this enzyme name is guanylyl. Sorry, that is guanylyl cyclase, and you can also call it guanylate cyclase. The same thing, guanylyl cyclase or guanylate cyclase. So that guanylyl cyclase will be activated by the nitric oxide. Then what will happen? It will produce cyclic GMP from GTP. Okay, so from GTP, cyclic GMP will produce by the help of guanylyl cyclase, and that guanylyl cyclase will be activated by the nitric oxide. Okay, then what will happen? That guanylyl cyclase, cyclic GMP will activate the PKG, that is protein kinase G. That protein kinase G will activate another enzyme that is called phosphatase. And this is indirect activation. One protein is there. That protein is inhibitory protein of phosphatase, and that inhibitory protein will be inhibited by phosphorylation by the PKG. So phosphatase will be activated indirectly by the PKG. That's phosphatase. You know the function of phosphatase. The phosphatase enzyme function is dephosphorylation. Okay, so here this is the myosin. Myosin, and this is the phosphate. This is phosphate. So this is active myosin, because this is the phosphorylated myosin. And what is the function of phosphatase? Dephosphorylation. Okay, so what will happen? By the help of phosphatase enzyme, that phosphorylated myosin will be dephosphorylated. That means phosphate group will remove. Okay, so that myosin will be inactive because this is not phosphorylated. So free myosin, and free myosin is not able to bind with the actin. Okay, so when in smooth muscle, when myosin will bind with the actin, or actin bind with the myosin. When myosin will be phosphorylated, then it will be able to bind with the actin, and that will form a actin myosin complex. But already myosin is dephosphorylated by the phosphatase enzyme, so no actin myosin, no bind with the actin myosin, not bind with the actin. So you can say no actin myosin complex, no actin myosin complex. Okay, or this is no, this is not for NO. You can show in that way, no nitric oxide, no actin myosin complex. And you know, for the muscular contraction, that can be skeletal muscle, or that can be smooth muscle, or that can be cardiac muscle, actin myosin complex is very much important. So here no actin myosin complex, because myosin is already dephosphorylated in active form. Then no actin myosin complex means no contraction, no contraction, no contraction of the smooth muscle, that is blood vessel. So finally, no contraction means relaxation, that means vasodilation. Okay, so this is the pathway. This is the pathway of vasodilation by acetylcholine. This is the parasympathetic pathway. You know, parasympathetic pathway vasodilation occur, and sympathetic pathway vasoconstriction occurred by the adrenaline. Okay, noradrenaline. But here parasympathetic pathway acetylcholine. Okay, so already I talked about another things, that is histamine, that is nitric oxide, and how they are connected with that pathway. Okay, so just I will try to summarize it. Acetylcholine bind with the muscarinic receptor 3, and that is G protein coupled receptor, and that G protein will be activated, that is GQ, and then that will activate the phospholipase C beta enzyme, and then it will produce IP3 and DAG, that is the second messenger. And here the IP3 bind with the endoplasmic reticulum, and endoplasmic reticulum will release calcium, and intercellular calcium will increase, and that calcium will activate the eNOS, that is endothelial nitric oxide synthase, and that will produce nitric oxide from the arginine, and that nitric oxide will just cross the endothelium and come into the smooth muscle of the blood vessel and bind with the, activate the enzyme, that is guanyl cyclase. Okay, that is guanyl cyclase, and that guanyl cyclase will just convert the GTP into the cyclic GMP, and that cyclic GMP is another second messenger, and that will activate the PKG. The PKG is another protein kinase G, and that will just phosphorylate the inhibitory protein of that phosphatase. That means indirectly it will activate the phosphatase, and when phosphatase enzyme will be activated, and the function of phosphatase enzyme is dephosphorylation. It will just dephosphorylate the myosin, and when myosin will be dephosphorylated, then it will be inactive, that will free myosin, and it will not able to bind with the actin. No actin myosin complex, no contraction, that means relaxation, that means vasodilation. Okay, this is the common pathway. And if you think some in your body somewhere inflammation occur, and one of the major inflammatory mediators is histamine. Okay, so if you think this is the blood vessel, and blood vessel surface is just carrying a histamine receptor H1. Okay, so what will happen? H1 receptor is also a G protein coupled receptor, and also this G protein is also GQ, that is GQ coupled receptor on the surface of the blood vessel. Okay, so what will happen? When histamine will bind with the histamine receptor, and G protein will be activated, and that same pathway vasodilation occur. But histamine will produce when? During the inflammation. Okay, so inflammatory vasodilation. Okay, so in that way, if you think If you take in emergency cardiovascular problem, that is cardiac problem, you can say as like mild heart attack or angina like, then we usually take nitroprusside or nitroglycerin. Why you will take nitroglycerin and nitroprusside? Then nitroglycerin and nitroprusside will just convert into the or produce nitric oxide, and this is the master molecule of vasodilation. Okay, and then it will just follow the pathway to causes vasodilation. Okay, so how come nitric oxide? So nitric oxide come from your blood vessel endothelium. This is endothelium from here, endothelium. Okay, endothelium, and also it can come from your some food. It can come from your pharmaceutical product, that is drug, and also nitric oxide can produce from endoplasmic produced from endothelium, but it can, your RBC can carry nitric oxide. Okay, so in the multiple way vasodilation can occur. Okay, so this is the uh, basically the simple explanation of the vasodilation. Okay, so thank you very much.